I want to turn it over to Jill, who is really going to talk about how this, how this all impacts families with autism, and thinking about, in addition to intervention, a model of potentially, prevention. So go ahead, Jill, here.
I'm very grateful to have this opportunity to be speaking with you about our symposium and share with you some information that I think is highly relevant to autism and may, in fact, shed some light on the increasing rates of ASD and other neurodevelopmental abnormalities we're seeing today. The idea that comes from epigenetics that may be very relevant to autism, actually there are several ideas from epigenetics, but this is the one we'll be focusing on here, is that epigenetics is like this fine-tuning of our genetic code, but what happens when that fine-tuning process goes awry, when the instruction book is damaged in some way? It could be that novel exposures from our recent past have impaired the development of our germline, which is, of course, our sperm and egg, contributing to the rising incidence of pathologies that we're seeing in our children. It's a really simple idea, actually, and it dovetails pretty well with what we've been seeing from ASD research. We see a lot of evidence for heritability of autism, but we don't see a lot of evidence for this Mendelian genetic kind of inheritance coming from previous generations. We see a crazy quilt of kind of de novo mutations and changes in gene expression in children that's really perplexing and mysterious. And so that part really isn't new, but the part that is kind of new is that exposures that don't go back very far in history may have something to do with some of these changes that we're seeing in a subset of autism. Obviously, I'm one of these people who believes that autism has many, many causes. We're just talking here about one potential subset. So I'd like to talk about these environmental exposures. When we think about environmental exposures, we often think about smog, pollution, water pollution, endocrine disruptors like pesticides, BPA, other toxic chemicals. But there's a whole other class of exposures that I think has been wholly overlooked, not only in autism research, but in many other fields of research. And that is this what I call the prenatal pharmaceutical craze of the post-war era. From the 50s and even through to today, but mostly kind of peaking in the 60s, there was an incredible enthusiasm for the use of synthetic chemicals marketed as pharmaceuticals for the prenatal market. And this is just an example of 4 categories of these kinds of drugs: the synthetic hormone drugs, sedatives, hypnotics, anti-nausea drugs, weight loss drugs. There were many others. And none of these were benign, and some of them were in fact quite catastrophic. You can see thalidomide is one of them. I obviously don't have to tell you that story. DES was another. Many millions of fetuses exposed to DES, causing, and now we know multi-generationally, pathologies in the affected offspring and even the grand offspring. These were novel exposures with no biological precedent in millions of years of mammalian or human history. And I think even today we haven't really come to grips with what these have done. So let's look at a quick snapshot of drug use from this era. This is from a very large cohort called the Collaborative Perinatal Project of more than 50,000 pregnancies. And the point of this is to draw your attention to the fact that a substantial percentage of pregnancies involved use of drugs, most of them synthetic, some of them natural. And this study showed that about half of the pregnant women in this time period were found to have taken at least 2 to 4 drugs during early pregnancy. That said, drug use varied widely by geography, by demographic, by socioeconomic status, and even by medical practice. So why does this history matter? This was a long time ago. I'm talking about like the 50s and the 60s and 70s, and like here we are in 2013. That stuff happened a long time ago. Why would all this old news matter to us today? I think it matters because the science is now raising some really strong red flags about the multi-generational effects of exposures. This is just three examples. I could have had 10 slides on this, but We now know that endocrine disrupting chemicals and other chemicals can induce molecular epigenetic changes such as DNA methylation and histone modification. There are many layers of epigenetic changes that could happen, but those are two that are best studied. We know that the epigenome is most vulnerable during early development. Now I'm sorry to get graphic here, but when did our eggs develop? We were born, all of us ladies in the room, we were born with all of our eggs, right? Our eggs developed basically in the first half of gestation when we were, this big, when we were tiny. And for males, it's very similar, different process of reproductive cell development, but you're born with all of your spermatogonial stem cells, which then go on to become your mature sperm later in life after puberty. But the most vulnerable period is that period when we're this small and our germ cells are developing. We also know here at the bottom that endocrine disruptors have been shown to exhibit transgenerational effects that are relevant to ASD, and that's from animal models. So we're seeing a lot of hints about why this stuff may be very important. Now, we just talked about the second point, that the fetal germ cells are very vulnerable, but that's not the only period of vulnerability. Preconception, fertilization, the early embryo, there's epigenetic vulnerability, and also spermatogenesis through the male's lifetime. And there are others. Another point is that the exposure need not coincide with the window of susceptibility. There are some chemicals that stay in our fatty tissues forever. So you might have an exposure when you're 10, and then you might have a kid when you're 30, but that exposure might still be relevant. It's pretty complex. So, as Alycia mentioned, and as I just talked about, the pregnancy exposures can affect 3 generations at once. And this is really a paradigm shifting idea, because now we know that exposures that happened a while ago can have downstream effects affecting grandchildren or even later generations. We also know that somatic cells, all of our body cells, are affected a little differently than our germ cells, and that you may see, in fact we do see this in animal models, different effects of exposures in different generations. There's also this idea of this latency period, that if you're exposed to something in utero, you may have this latent birth defect of, in your germ cells, but that doesn't manifest until your kids are born. All right, so what does this mean? Does this have any relevance for autism? I definitely think so, which is why I'm here. And I'm going to offer myself and my children as case study number 1 in this multi-generational hypothesis. I was born in 1965 in Los Angeles. I still have the same haircut. And I developed, I would say, pretty normally. Some people might dispute that, but I went on to graduate from college and law school, and pretty successful in my careers, so kind of normal. There's no autism or developmental or psychological conditions in my ancestry or extended family or in my husband's. We had 3 kids, and everything was normal. Normal conceptions, normal pregnancies, normal deliveries, no unusual exposures, no genetic anomalies, and we even did microarrays looking for copy number variations, deletions, nothing. They're physically gorgeous. They're very robustly healthy, wonderful kids. But 2 of them have highly incapacitating abnormal neurodevelopment, which has been labeled idiopathic autism, basically saying, we don't know why they're so impaired. But as much as I love them, they are by any reasonable measure catastrophically neurodevelopmentally disabled, and they're going to need lifelong 24/7 care. So why did this happen, and why might this be happening to a subset of kids kind of like them? Well, here's an idea. Very recently, through a succession of complete miracles that I really don't have time to relate at the moment, I stumbled upon some very shocking information, and that very shocking information was very detailed records of my prenatal exposures from 1965, from when I was that big. To say that obtaining these kind of records is rare is an understatement. I always ask, so I'm going to ask it now, has anybody in this room ever seen their prenatal records? No. I mean, I still haven't found anybody else. I don't know. Why me? I don't know. But these kind of landed on my lap. And it took a while, but with the help of a lot of researchers and physicians, I was able to decipher them. What do these records say? It said that I had been exposed to a veritable barrage of synthetic steroid hormone drugs from the very beginning of pregnancy through at least the 2nd trimester. why was I exposed to all of these fake progesterones, corticosteroids, fake estrogens, and maybe a couple of other things? Why? This was 1965. What happened then that maybe isn't happening now, or maybe happening differently now? What happened back then is a couple things. First of all, this category of drugs were used very, very heavily in pregnancies that were considered at risk, or where the doctor considered the woman to be a habitual aborter. That was a term that was prevalent in the medical community at the time. A habitual aborter was any woman who had 2 or more previous miscarriages. I hope everybody knows about DES, which is one of these quote-unquote anti-miscarriage drugs that didn't really work. I was not exposed to DES. The drug that was actually more prevalent were these progestins, these fake progesterone drugs, and I was exposed to 2 of those in addition to these estrogens and corticosteroids. It was a prednisone kind of drug that I was exposed to. So why should we care? And also, some people say, well, Jill, is that a lot? And you were exposed to about 5,000, 6,000 milligrams of these synthetic hormones. Was that a lot? Well, I'd like to compare that to birth control pills today. A birth control pill has about 0.18 milligram of synthetic hormones in it. If you were to add up a year's worth of birth control pills, it would hardly register a blip on this graph. So I was very heavily exposed. In fact, the researchers at the time marked on anyway, small tangent, I had been researched, I didn't know this, when I was 8 years old, because I had been so heavily exposed to these drugs, which is part of the reason that I found these records. So I just found out very recently that I had been a subject matter of research study. But this was a massive amount of exposure. I mean, really, it's amazing that I'm kind of quasi-sort of normal, I think, but whatever. We won't talk about that. So yes, did these drugs affect me? I think they did. There's reasons to believe they did. But moreover, did they affect my eggs? And given that millions of people were exposed to these drugs, and tens of millions were exposed to many other kinds of endocrine disrupting drugs, these are overt endocrine disruptors, but other drugs were had endocrine disrupting effects as well. Did it affect our germline, or is the germline impenetrable? Was it impermeable? Did it not change, or was the effect negligible? Well, the answer is we need to know more. I don't pretend to have all the answers today. Clearly, I think that this is an interesting and intriguing idea. But we need to know more. We need to consider the possibility that some environmental factors may have wreaked havoc on our germ cells. I think that the prenatal pharmaceuticals are, of course, just the tip of the iceberg. This is Mad Men era, right? The 50s and 60s and 70s. We had DDT, we had dioxin, flame retardants, PCBs, talk about persistent pollutants. And many other candidates. Maternal smoking was at its apogee in this era. Recreational drugs, air pollution, nuclear testing even, Superfund sites, and I would even throw in birth control pills, since those are also endocrine disruptors in and of themselves. So there's a lot to think about in terms of exposures that could be having multi-generational effects. We run a little philanthropy, an autism philanthropy. We've already kickstarted some projects just to try to get this hypothesis examined and try to get it off the ground. We picked some low-hanging fruit, so to speak, and have, with Autism Speaks, funded the first epidemiological study in Denmark looking at 3-generation effects of smoking and prenatal pharmaceutical use from the 60s. We are interested in prevention, and one thing that's very near and dear to me is the idea that we know there's really quite a bit of consensus that prenatal pharmaceutical use can affect human germline, but never has that been on the radar of the FDA. There's no testing protocol. There's no warning. It's not part of any way to assess systematically risks of exposure. So we're trying to get the germline, our germ cells, the most important cells in our body, on the radar of the FDA. We have the symposium. We're sponsoring some research into this history, which is really a very hidden history. We want to make medical records much more available to all Americans so we can all know. I don't want to be the only person in America who knows her own prenatal exposures. I want everyone to be able to know. And some laboratory studies as well. Alycia and I, and we're working with the symposium, came up with some recommendations, more broad recommendations for research. She already touched on it. I'm not going to go into it in detail, but we do need kind of a multifaceted approach to look at these ideas about how the environment may have affected our genes and may be having these downstream impacts. Thank you so much.